So for table C, it's going to be really important that you change benzaldehyde to acetaldehyde. And table C is where in the lab you would have done some chemical tests on various aldehydes and ketones. So the first thing that you're asked to do is determine the solubility in water of propanol, benzaldehyde, acetone cyclohexanone, and an unknown. You cannot make a prediction at this point on your unknown, but we'll look at where you can make a prediction later for your unknown, or be able to fill that in. So for these chemicals that are listed, make a prediction based on the number of carbons. So you may remember from your reading or from the video lectures that because ketones and aldehydes are polar, the smaller molar mass aldehydes and ketones are actually soluble in water. So for the chemicals that you know, predict their solubility. They have 5 carbons or less, they should be soluble in water. 6 carbons is right at the boundary, so maybe partial soluble or insoluble, and 7 or more will not be soluble in water. All right, the second part of Table C is the iodoform test for methyl ketones. And so again, you're just going to predict whether an iodoform test should be positive or not. And an iodoform test, if you remember from reading in the beginning of your lab, an iodoform test allows you to know if a methyl ketone is present. So that means that a methyl group is bonded to the carbonyl group. So you are going to predict for everything except your unknown whether whether an iodoform test should be positive or not based on the structure. All right, and then Table C, Benedict's reagent. You remember from video lecture as well as from the reading of the lab that Benedict's reagent is a mild oxidizing agent, and it gets reduced and changes its color in the presence of an aldehyde. So again, remember to change benzaldehyde to acetaldehyde. And then the chemicals in the photos, it may be hard to read the labels, but they are organized according to the directions. So on the far left is propanol or propionaldehyde, then comes acetaldehyde, then acetone, cyclohexanone. And the unknowns are in a separate photo. So you're going to use the photos that are found on this page a little bit lower to answer the questions about Benedict's reagent. So here is Table C. Your solubility is going to be based on the structure. The iodoform test is again going to be a prediction based on the structure. And so this methyl ketone, yes or no, really you're not going to be able to answer to. It's just predict, predict, predict. And then this is sort of a reiteration of column 2. And this is a little confusing here where it says color. Basically, this is the color of the Benedict's test. So it's either going to be green, blue, or orange. So here are the photos of the Benedict's test, and from left to right are the chemicals. And then here are photos of our lab tech doing the actual chemical reaction. Let me see if I can get that. So here, after the chemical is placed in a water bath for 5 minutes, if there is an aldehyde, then the Benedict's reagent will oxidize it, and the Benedict's reagent gets reduced and it forms an orange color. And so we can kind of see this changing of color, even though it's not completely orange yet, means that there is an aldehyde in that, an aldehyde functional group in that chemical that's being tested. And so here are the results. And then I just want you to notice here that the Benedict solution is an aqueous solution. And so here we see a separation right here of the cyclohexanone. And that's sort of what we would expect. Cyclohexanone has 6 carbons. That's really at the edge of the solubility of aldehydes and ketones in water. And so it's not completely soluble, as we can see from this layer on the top here. All right, so you have 2 unknowns that you're going to have to fill out. Here are the Benedict's reagent results from unknown 1. Based on that and based on this appears one of these four chemicals, you should be able to identify it. And then unknown 2, you should also be able to identify not on the color of the Benedict's solution, but on its solubility.